Good morning, everyone. Good morning. Happy Wednesday. Good to see everybody. Okay, still waiting for a couple more people. It looks like everybody got their lecture exam in. Excellent. I'll be grading those today and maybe into tomorrow. So keep an eye out for your score on that. That was your last lecture exam before the final. This week, you're looking at an introductory lecture to immunology. You'll be watching videos related to the immune system and how the immune system functions. Today is actually the last lab day that we will be talking about biochemical testing. Today we're going to be talking about respiration. And that means that next week in lab, we're going to be spending a little time on Monday talking about your final lab assignment for the course. And of course, I'll just introduce it today, but you're going to be receiving a data set. You're going to be receiving a list of test results from the microbiology lab about a microbe that was in a sample that was sent to the lab for identification. So you're going to have a chance to apply all of the things that you've been learning this semester. You're going to have a chance to interpret a list of test results, and then using some tools that I'll provide for you, you'll have an opportunity to identify this microbe that was sent to your lab into your capable hands for identification. So we'll talk about that assignment on Monday, take a look at it, and then that's what you'll work on next week, which is the last full week of our course. You also have on Wednesday next week, you have your final lab practical. So all coming up very quickly. I do want to point out, because I forgot to mention it when we were together on Monday, you have two separate lab assignments this week. One is listed as lab questions for protein catabolism, and one is listed as lab questions for respiration. Those are the final two 5-point laboratory homework questions. So yeah, there are two of them. Gives you a chance to earn a few more points. Neither one of them is particularly long. So I decided to split them and again, give you a chance to earn 5 more points. Those are due on Sunday as usual. Remember, if any questions come up while you're watching the immunology material, feel free to message me or to post questions on the discussion board, questions. Remember, sometimes your classmates are a good source. So do take a look at that board from time to time and see if you can help one of your classmates answer a question or two. All right. Just about everybody's here now, so I'm going to go ahead and get started and pull up our slides for today about respiration. And that's what you should be seeing on your screen. The title slide that says Lab Respiration. So the first thing I want to do this morning is just review this very complicated information we've been learning about regarding metabolism. First, remember that that word metabolism is nothing complicated. It just means all of the chemical reactions that occur inside a living cell or a living organism that keep that organism alive every day. It's the sum of all of the reactions. When we talk about oxidative metabolism, remember the word oxidative has nothing to do with oxygen. Instead, it refers to oxidation reduction reactions. Remember, there's a whole group of chemical reactions that are referred to as oxidation reduction reactions. They occur all over nature, including inside human cells and other cells, not just humans, other cells, including microbial cells. Oxidation reduction reactions get that name because two processes are going on. Something, some molecule, some atom within a molecule is losing an electron. And that means some other atom in a different molecule is gaining an electron. So the electron is moving, it's shifting, it's transferring. Remember, oxidation refers to the process of losing. And we remember that by saying LEO, lose electron oxidation. Reduction is the process where something gains an electron. And we remember that by the term grr. So Leo the lion goes grr. Oxidation reduction. These reactions, these oxidative metabolic reactions include the way that cells catabolize or break down carbohydrate. When a cell breaks down, for example, glucose, it does it through a long series of oxidation reduction reactions. So we talk about the oxidation of glucose. Remember, not about oxygen, but about taking electrons out of glucose. And of course, a cell does that by breaking the covalent bonds in glucose. Because all a covalent bond is a pair of electrons that are being shared between two atoms. So if you want to set those electrons loose, you got to break that bond. Now, why would a cell want to break all the bonds in glucose and set all those electrons free? The answer is that there's energy to be harvested out of electrons. Electrons are the conduits of energy. So by setting all the electrons free from glucose, we can send those electrons down the electron transport chain, and specialized proteins and other molecules are going to harvest that energy. And then they're going to do some critically important work with that energy. And the work is making ATP. The energy is used for a process called oxidative phosphorylation. Again, oxidation reduction. Oxidative phosphorylation. The thing that's getting phosphorylated is ADP. We're going to phosphorylate it. We're going to put a phosphate group onto it and turn it into ATP. Now, how does the cell manage that? Well, it uses the energy from all those electrons that came out of glucose. It uses that energy to send hydrogen ions out of the cell, to pump them out of the cell. And in microbes, that's into the periplasmic space and build up a big gradient of hydrogen ions. Then that gradient is allowed to resolve, to go away. All the hydrogen ions come right back in to the cytoplasm of the cell by flowing through a very ancient molecule called ATP synthase. This molecule has been on this planet since the beginning of time, since the beginning of life on this planet. All living cells have this molecule. Isn't that fascinating? I know, I'm a terrible nerd. Hydrogen ions flow through a channel in ATP synthase back into the cytoplasm. And as they flow, they literally cause a spinning motion in the molecule. And that spin, that mechanical spin, is what drives the phosphorylation of ADP into ATP. It's an extremely efficient way to make ATP. From one glucose, you get somewhere in the neighborhood of 30 ATP produced. Very, very efficient. Oxidative metabolism. We know that microbes can make ATP in this way using oxidative metabolism. But what's especially interesting about them is that they can do this oxidative metabolism in two different ways: aerobic and anaerobic. Some microbes do cellular respiration and oxidative phosphorylation the same way we do. In other words, they use oxygen as the final electron acceptor at the very end of the electron transport chain. Remember, what's happening in the electron transport chain is the gathering of the energy from the electrons. Those specialized proteins are pulling the energy out of those electrons, but the electrons themselves are going to end up at the bottom of that staircase that we call the electron transport chain. And they're going to need to be received. Glucose gets oxidized, something else has to be reduced. And at the end of the electron transport chain, in aerobic organisms, that's oxygen. Oxygen receives the electrons, water is produced, all is well. But microbes are much more diverse metabolically than humans are. And so there are microbes who do this long oxidation reduction reaction using a different final electron acceptor. There are lots of these in nature, these molecules that are able to sit at the bottom of the electron transport chain. But in anaerobes, there are three that are the most common. First is nitrate, then is sulfate, then is carbonate. These are oxygen-containing molecules, and they can receive electrons. They're able to be reduced in this way. Some microbes do cellular respiration. Some do it aerobically, some do it anaerobically. Very, very diverse set of organisms. Now, something else to remember about cellular respiration and oxidative phosphorylation. The bulk of the ATP is getting made at the end. That makes sense, right? We go from cellular respiration, which is technically, from the moment the glucose first begins to be broken apart until the end of the electron transport chain when the oxygen receives the electrons. That's the bulk of cellular respiration. Oxidative phosphorylation then follows when the proton gradient is made, when it flows back through ATP synthase, and ATP gets built. So one follows the other. But we do need to remember that a little bit of extra ATP is made in the midst of cellular respiration. A little bit extra gets made. A little ATP gets made during the process of glycolysis, which is the first set of reactions in cellular respiration. Now, that ATP is not being made by oxidative phosphorylation. That ATP is being made by substrate level phosphorylation. Later on in cellular respiration, remember, we go through glycolysis, then we go through pyruvate processing, then we go into the Krebs cycle. During the Krebs cycle, we get a little more ATP from substrate level phosphorylation. So these oxidative reactions, cellular respiration and oxidative phosphorylation, most of the ATP is made through oxidative phosphorylation, but a little bit of extra is made using substrate level phosphorylation. This happens in both the aerobic version and the anaerobic version. We know that there is another way for cells to make ATP outside of breaking glucose down in cellular respiration. There's a whole different pathway available called fermentation. Some microbes can do it, some microbes can't. And of course, there are some microbes that only do fermentation. They never do cellular respiration. In fermentation, ATP gets made, but it's made using substrate level phosphorylation, much less efficiently. But ATP is ATP. So some microbes use just fermentation. And of course, there's a huge number of microbes out there that combine these things. They can do different pathways depending on the environment they find themselves in, depending on whether or not oxygen's available, or whether or not nitrate's available, or whether or not they're able to ferment. Some microbes can do a combination of pathways, and they switch back and forth depending upon the situation, the circumstances, the environment. They're much more diverse than we are. If you and I don't have ready access to oxygen in the air that we're breathing, we don't have long to live. Imagine how great it would be if you found yourself in an environment with no oxygen, or let's say you had a heart attack and you stopped breathing and it was perfectly fine because your cells would just switch and do something else. We're not that lucky, but the microbes are. The microbes are. Now I'm going to put another review point right here, and this is an important one. I wrote this down as a comment in the homework assignment for many of you this past week. Living cells use carbohydrate for all of this that we've been talking about. Carbohydrate is the macromolecule that living cells use to make ATP. Carbohydrate has the energy within it. Remember, when you hear that word energy, think electrons. Carbohydrate has the energy in it, to fuel the metabolic reactions and make ATP. Now, sometimes that's a hard thing to really take on board, that it's carbohydrate, because in our own human lives, we think, well, I'm also getting energy from protein and fat, right? Well, you are and you aren't. Carbohydrate is the molecule that cells use, all cells, to make ATP. It is possible for your cells to use other macromolecules to make carbohydrate and then break it down to make ATP. But it's carbohydrate that is the fuel for making ATP. When you tell some people that, they get very surprised, especially in days like today when a lot of folks who are trying to lose weight are doing things like the keto diet, where they're eating very small amounts of carbohydrate, very, very low levels of carbohydrate. And you might say to yourself, well, what about those folks? I mean, aren't they using protein to make their ATP? And the answer is no. No, they're not. They're still using carbohydrate. But because they're not eating their carbohydrate, their body has to go through extra work to make it. There's a process called gluconeogenesis. How's that for a word? Gluconeogenesis. And if you break that down, it means making new sugar. And your body is able to do this, but it's a very difficult metabolic reaction, and your body would much rather not do it. Your body is built to use carbohydrate that you eat for energy. But yeah, if you get in a bind, or if you choose not to eat carbohydrate, your body has to manufacture it and then send it in to those pathways that will break it apart to make ATP. Remember, protein is used to build things, right? As enzymes, as receptors. There are so many uses for protein. But when our bodies break it down, it's usually because they're trying to get at the amino acids. Our bodies break down protein most of the time to get the amino acids out so that we can make new proteins. That's how important proteins are. But no, proteins aren't used to make ATP. Carbohydrates are. Fat, the other macronutrient, fat is for storage of electrons. And again, fat can be oxidized, fat can be broken down, just like all macromolecules can be broken down. And the human body is going to do that when it needs to get ATP from carbohydrate. It's quite fascinating, isn't it? So Connie is talking about doing a very restrictive diet that had essentially no carbs in it, not even fruit. Ugh. Oh, Connie, I don't even think I could do that. And you were very tired. Yeah. Talk to people who are on the keto diet. Or if you've been on it yourself, you're going to experience what some people sort of flippantly call the keto flu. You're going to feel lousy for several days, maybe even a week. And that's because your body has to completely switch from burning carbohydrate that you're eating into a whole new process of manufacturing carbohydrate so it can be burned. And it makes you feel lousy because it's not the way you were built to make ATP. There's another interesting thing about keto. You do tend to lose weight, by the way. It's hard work to be on a ketogenic diet. Here's the other interesting thing, though. The weight loss tends to occur relatively quickly, which is why people like doing keto. But it plateaus quickly as well. So the ketogenic diet is really only effective for weight loss for a period of about 3 months. At that point, you've gotten everything out of it that you're going to get in terms of weight loss. It's not a healthy diet to stay on long term because you're not built to do that. You can do it. Your body is capable of doing some amazing things, but your body was not built, structured to do that, and it is not healthy over the long term. That was what Chelsea was asking. No, Chelsea, it is absolutely not healthy over the long term. The other thing you have to remember about a ketogenic diet is that in place of all that carbohydrate that people are no longer eating, they're eating a lot of fat, aren't they? They're eating a lot of fat. 50, 60% of their calories are coming from fat. We know that that's not good for us as humans. That's not good for our cardiovascular system. That's not good for the general functioning of our organs. Even when you're eating primarily good fats, you can overload your body with fats. Your body was, again, not built to survive long term off of fats. So not a good idea for long term. I have a friend who is obsessed with the keto diet, says Elaine. Oh, I've met a few of them, Elaine. Insists she has never felt better. How is it possible if her body is having to work so much harder? Well, Elaine, I would suggest there is a little psychology in that I imagine your friend is losing weight. And the psychology of losing weight, especially for a person who has struggled to lose weight, can be very powerful. If your friend is down, say 10 or 15 or even 20 pounds, that's a big psychological lift. Number two, if you lose 10 or 15 or 20 pounds, your body is going to feel better, right? When people lose, I won't say significant, but decent amounts of weight, 5 and 10% of their body weight, just having that weight gone off of your skeletal system also makes you feel a lot better. It's easier to breathe. It's easier to move around. You tend to be more light on your feet, so to speak. And so, yeah, I imagine that people do feel quite good after they've lost weight on this diet. Here's the really sad news, though, and you probably already knew this. People who lose weight on the ketogenic diet and then return back to their regular diet 3 months down the road, maybe 4 months, guess what happens? They tend to put all that weight back on, and they often tend to put more on than where they started. This is true of all diets that are designed for short-term weight loss. The human body is really, really good at losing weight in the short term. All you have to do is restrict your calories or do something like the keto diet. You can lose weight. The problem is keeping it off. Because once a person goes back to a more traditional diet, all of their bad habits tend to return. The best way to lose and maintain that loss is changing the way you eat fundamentally. And most people struggle to do that. It's very hard. It's very hard in our society, particularly because we're surrounded by terrible cues to eat all day, every day. Yeah, good questions. Elaine's asking, if a person stays on the keto diet long term, what would that do to the body over time? Elaine, some people do stay on it. I don't know why you would stay on it because, again, your weight loss will plateau. But maybe some people say, well, I lost my weight on keto, so I'm going to keep doing keto. Here's why that's really dangerous. Keto can be done wrong. And sometimes people get very extreme about it. The reason we call it keto is because when the human body and other animal bodies break down large amounts of fat, and that's what's happening during a ketogenic diet. a waste product is produced called a ketone. So when you're on the keto diet, you want to start making ketones. In fact, people check their urine with dipsticks to make sure that they're in ketosis, as it's called. They are making ketones. That tells them that their body is burning fat. Ketones are toxic. I always have to remind people, by the way, ketones are toxic. And so, yes, when you start burning fat, you're gonna lose some weight. But remember, it's not good for your body over the long term. And there have been people who've gotten into real trouble on the keto diet because they took it to an extreme. They got their carbohydrate levels so low and their ketone level so high that they ended up in the hospital because they can go into an acidotic state. Their body pH can change. And that's not an easy thing to do to your body, by the way, but it'll happen. So if you were under a doctor's care in that your ketone levels were being monitored so that you made sure not to go below a certain level of carbohydrate. If you did it by the book, you could stay on it under a doctor's care. But again, I don't know why you would, because it's not nutritionally sound. The amount of fat is too high, and the weight loss doesn't keep happening. You plateau on your weight loss. So again, I don't know why you would. Yes, John, you would have horrible cholesterol because unfortunately, the ketogenic diet, at least most versions that I've read, does not distinguish between animal source fat and plant source fat. And of course, it's animal fat that contains cholesterol. If you're eating 50 to 60% of your diet as fat, and you're eating a lot of animal fats, your cholesterol is going to get really high. It's not good for your cardiovascular system to eat that much fat, at least not for the long term. Listen, there's nothing wrong with the ketogenic diet for a few weeks if you want to try it and you want to see if you can lose some weight. But statistically, you will not keep that weight off. Statistically, you won't keep that weight off. It's sad, but it's true. There's no magic diet. The only magic is in controlling what you eat, keeping it as natural as possible, eating whole foods, natural foods, limiting how many calories you take in every day. It's very hard work, very, very hard work. And most people aren't willing to do that. Most people want a quicker fix. It's hard being human. Connie, I totally agree. Oh, Elaine's asking about medical uses of keto. Yeah, there are some medical uses of keto, and it's a little more complex, Elaine, than I can get into right now because of time. But yeah, there are reasons, there are certain medical reasons to limit how much carbohydrate a person is eating. And if you can instead switch over onto a ketogenic diet, those people, you're talking about seizure disorders, those people can be managed better on a different diet. But again, it's done under a doctor's care. So they're getting their blood work done, every couple of weeks so that they're being very careful about how many ketones are they're allowing in their blood. But yeah, it's interesting, isn't it? Dietary manipulation can really help in some very serious, significant medical disorders. Yeah, that's going to be my quote of the day, Connie. It's hard being human. It's very hard being human. All right. Enough about carbohydrates. Actually, we're still talking about them because we're talking about respiration today. All right. So I mentioned last time that as we talk about carbohydrate catabolism today, we're going to focus primarily on anaerobes. But we're going to look at two different tests, and one of them keeps us with the aerobes for a minute. Remember, aerobes use oxygen as their final electron acceptor at the end of the electron transport chain, and that oxygen is going to get reduced. The glucose is what's being oxidized. All of the electrons from the glucose are being removed. The oxygen molecule is being reduced, and water will be produced as the product. The pathway of reduction, in other words, the reaction at the end of it, is that oxygen molecule receiving electrons and becoming water. Now, one thing I didn't put in this equation is not complete, because of course you would also have to add hydrogen in there. But essentially, oxygen is being reduced with electrons, and you're getting water made as the product. Now, in the electron transport chain, there are 4 complexes of specialized proteins that do the work of harnessing energy out of the electrons as they pass by, as they pass down the staircase. There are helper molecules in the electron transport chain that are passing the electrons from one complex to the next complex. In lecture, we talked about a molecule called Q or ubiquinone. And there's another molecule called a cytochrome. And I have the word highlighted here. Cytochromes are the little helpers too in the electron transport chain, in that they help move electrons from one protein complex to the next. There are 4 different protein complexes in this staircase that we call the electron transport chain. And the electrons have to travel through each one of those complexes in order to get their energy harvested. The protein complexes can't just hand electrons to each other. They need a middleman. They need a messenger. So the first complex hands the electrons off to the cytochrome delivers them to the second protein complex. Then the second protein complex hands them to cytochrome, and cytochrome hands them to the third one, and so on. Ubiquinone does this work, and cytochrome does this work. It turns out that in bacteria, there are 4 classes of this type of molecule called a cytochrome. There's cytochrome A, B, C, and D. We're going to focus on cytochrome C today because we can test to see which type of cytochrome a species of bacteria has. And cytochrome C is going to be meaningful to us, as I'll explain in just a minute. Now, the anaerobes are very different because the anaerobes don't use oxygen as their final electron acceptor. They can use lots of different molecules. The most common final electron acceptors in anaerobic microbes are nitrate, sulfate, and carbonate. So most of the microbial species, particularly bacteria, are going to use these molecules. These are inorganic molecules. In other words, they don't have any carbon in them. But these 3 are not the only ones. There are others. There are some organic ones. There's one called fumarate. There's one called DMSO. There's triethylamine. Don't worry about those. Just know that the top 3 are nitrate, sulfate, and carbonate. Let's look at these pathways of reduction. If we start with number 2, when sulfate receives electrons, it's going to produce water just like oxygen does, but it's also going to make hydrogen sulfide. When carbonate receives electrons, it's also going to make water just like oxygen does, but it's also going to make methane. So there are pathways as in terms of what happens to these final electron acceptors once they receive all those electrons. Now look at nitrate. nitrate looks a little complex. And that's because there isn't one pathway of reduction for nitrate. There are actually three. But here's the kicker. They're not three separate reduction pathways. It's one long pathway. In other words, nitrate can be reduced this much, or it can be reduced this much, or it can be reduced this much. There are levels of reduction. In other words, sometimes nitrate takes on this many electrons. Sometimes it will receive this many electrons, and sometimes it will receive this many electrons. It all depends on the species that you're looking at. It's one long pathway of reduction, but we can describe it occurring in 3 steps or 3 parts. So let's take a look. When nitrate receives electrons, it becomes what's called nitrite. That's the NO2, and water. Water's always involved here. So nitrate gets reduced to nitrite. Now, sometimes that's not going to stop at that point. Sometimes this is all that happens. The nitrate becomes nitrite, we're done. But in other species, the nitrite will go on and get further reduced. It will accept more electrons, and you'll get a molecule called nitrous oxide. I'm sure you've heard of nitrous oxide before. And in some even further species, the nitrous oxide can get further reduced. You can add electrons to nitrous oxide, and what you're going to get is nitrogen gas. So when nitrate becomes fully reduced all the way, when it has taken on as many electrons as it can possibly take on, you're left with nitrogen gas. But some species don't take it that far. Some species will take that nitrate and reduce it to nitrite and then stop. Other species will take nitrate, reduce it to nitrite, and then further reduce it to nitrous oxide. And still other species will take the nitrate, reduce it to nitrite, further reduce it to nitrous oxide, and then further reduce it to nitrogen gas. So nitrate is really a fascinating final electron acceptor because it can take on different amounts of electrons. It can be reduced in stages, which is kind of interesting. You can imagine that, this might be a good thing to know about a molecule. About a bacterial species. If a bacterial species stops at nitrite, that would be a good thing to know because that's a trait, that's a characteristic of that organism. It might help us identify that organism in the laboratory. It would be good to know just how far in the reduction pathway does a particular species take that molecule. It might help us identify that molecule. Oh, Elaine's asking a great question. Is there a difference between nitric oxide and nitrous oxide? I'm not sure if I don't even know if there is a nitric oxide. We're going to have to Google that one, folks. Nitric oxide. Elaine, if there is it or if they're related, I don't know. I know that it's nitrous oxide that is produced in this reduction pathway. We'll have to Google that a little bit. All right. So we're going to do 2 tests today. The first test is the simpler test, and it's called the oxidase test. And what this test looks for is that little helper messenger molecule that we find in the electron transport chain, called cytochrome C. But this time we're looking for cytochrome C oxidase. Put a star next to this in your notes. This is a place where students frequently get confused. The molecule in the electron transport chain that helps move the electrons along is called cytochrome C. But there's an enzyme called cytochrome C oxidase that is what we're testing for. We're testing for the enzyme, not the actual molecule, not the cytochrome C molecule. So this test looks for this enzyme, and that's because this enzyme is needed for cytochrome C to do its job. So we're not actually detecting the cytochrome C, but we're detecting an enzyme that lets cytochrome C do its job. Now, how does this test work? Well, this is one of those really easy ones. This one is very similar in terms of its speed and ease to the catalase test. All you have to do is pick up some colony material on a cotton swab and drip onto those colonies a reagent, and it's simply called the oxidase reagent. When the oxidase reagent comes in contact with those cells, if they make cytochrome C oxidase, the swab is going to turn purple. Now, why do we care about cytochrome C oxidase? Why do we bother testing for that one? Well, it turns out that all organisms who use oxygen as their final electron acceptor also use cytochrome C. So all of the bacteria that use oxygen as their final electron acceptor, the version of cytochrome that they're going to use is cytochrome C. So it's a useful piece of information to know if a bacteria is positive on the oxidase test, because it means that they use cytochrome C and that they use oxygen as their final electron acceptor. Now, that's the first test we're going to look at. The second test we're going to look at is about nitrate reduction. This is going to test bacteria for their ability to reduce nitrate to nitrite. Remember, that's the simplest pathway to reduce nitrate. You can add electrons to nitrate and you will get nitrite. Now, the nitrate reduction test is also going to allow us to further investigate microbes that can take that reduction even further along. But the fundamental test is testing for the ability to reduce nitrate to nitrite. So remember that. This test uses a plain old nutrient broth. We've seen that culture media quite a lot. A plain old nutrient broth and a supplementation of potassium nitrate. We have to give the microbe nitrate if we want to watch it reduce that nitrate. So we're going to take our nitrate reduction test media. We're going to inoculate it, and we're going to incubate it, not for 24 hours, but for a full 48 hours, because it turns out that these reactions, especially the long reduction reaction, it takes time. So especially for bacteria that are going to take nitrate and reduce it all the way to nitrogen gas, we want to give them the time to do that. So 48-hour incubation. When you take the tube out of the incubator, you have to add reagents. We've seen this, right? We saw this in the VP test. We saw this in the phenylalanine deamination test. We saw this in the cysteine desulfuration test, adding reagents in when you take the tube out of the incubator. This test doesn't just add one reagent, it adds sequential reagents depending on what results you get. So this is the most complex test to conduct that we talk about in this course. Now, when you pull that tube out of the incubator, the first thing you're going to do is add 2 reagents. 2 reagents get added in right away, dripped into the tube, and we look for a color change to red. If the tube turns red, when you add those first 2 reagents in, you know that there is nitrate in there, and you have a positive test. The nitrate in the tube got reduced to nitrate, and the nitrate is what's giving us the red color with the combination of the reagents. So that's pretty good, right? If you take that tube out of the incubator and add in the first 2 reagents, if it turns red, great, you've got a positive test, and you can say this bacterium is able to reduce nitrate to nitrite. Now, what about if it doesn't turn red? We can't just say, oh, it's a negative test. If we put those 2 reagents in and the tube does not turn red, you can't stop there. What that means, if it doesn't turn red, is that there's no nitrite in that tube. But remember, some microbes take the nitrite and they keep reducing it. Some of them reduce it to nitrous oxide, and some reduce it all the way to nitrogen gas. So just because there's no nitrite in the tube at that moment, that doesn't mean that reduction isn't going on. It might mean that, but it might mean that further reduction has happened. So you can't stop. You got to add another reagent here. If you get no red color after you add those first 2 reagents, you're going to add a 3rd reagent. And this one is a very interesting one. You're going to add in zinc, essentially powdered zinc. What zinc can do is reduce nitrate to nitrite. In other words, if the bacteria that we grew in this tube was not able to reduce nitrate to nitrite, that would mean that the tube still had a lot of nitrate in it because the bacterium didn't reduce it. All that nitrate would still be in that broth tube. When you add zinc in, the zinc will take the nitrate and reduce it to nitrite, and the tube will turn red. All right? Remember, the red tells you that there's nitrite in the tube. If I add the 2 reagents at the beginning and the tube does not turn red, that tells me that there's no nitrite in the tube. All of the nitrate is still in there. How do I know for sure? Well, if I add zinc in and there's nitrate in the tube, the zinc will reduce it to nitrite and the tube will turn red. That is a negative test result. You might say, well, hold on. How can you have one red tube be a positive result and one red tube be a negative result? That doesn't make sense. It really does, though, if you walk it through. The red color means that nitrite is in the tube at that moment. If I put the first two reagents in and the tube turns red, that tells me there's nitrite in that tube, and it's the bacterium that did the work, that took the nitrate and reduced it to nitrite. If the tube turns red after the first two reagents, the bacterium did the reduction, took the nitrate and reduced it to nitrite. But if I pull that tube out and I put the two reagents in and I don't get a color change, that tells me, oh, there's still nitrate in here. The bacterium wasn't able to reduce it to nitrite. I can confirm that. I can confirm that if I put some zinc in. If there's any nitrate in that tube and I add zinc, the zinc is going to reduce it to nitrite. The zinc did that. The bacterium didn't do it. The zinc did it. That's a negative test result. The bacterium wasn't able to do the work, but the zinc did it. And the zinc showed us that the bacterium was not able to do it. That's a negative test result. Oh, we got a report back from Elaine. So nitric oxide has one less oxygen than nitrous oxide. Very good, Elaine. Very good chemistry for us this morning. So yes, so all I can tell you is that it is the nitrous form that is being produced during the reduction of nitrate. But thank you, because I had not heard nitric before. Thank you for that. All right. There is one more step to this test, because guess what? There's another possibility of what's happening. Imagine that I take the tube out, I put the two reagents in, and it doesn't turn red. So I take out my zinc and I put some zinc powder in, and it still doesn't turn red. What the heck is going on now? If there's no red color, even after you've put the zinc in, that tells you that the bacterium has not only reduced the nitrate, but it has reduced it past nitrite. It has either made nitrous oxide or it's made nitrogen gas. Now, how do we know the difference? The nitrous oxide is going to just dissolve into the medium, but the nitrogen gas doesn't. So what we're going to do is we're going to put a Durham tube in here. We're going to put an upside down Durham tube in here. When we add the two reagents and get no color change, and then we're going to add the zinc and we still get no color change, the next thing we're going to do is we're going to look in the Durham tube. If there's gas in the Durham tube, we know that the microbe has reduced the nitrate all the way to nitrogen gas. If there's no gas in the Durham tube, we know that the bacterium has reduced the nitrate to nitrous oxide. And now we're finally done. So I've put in a couple of extra slides in this set because I know how complex this pathway is. So I've tried to summarize the test a couple of different ways, hoping that one of those ways will make sense to you as you're learning the material. So when we do the nitrate reduction test, when we take the tube out of the incubator, because remember, we have to inoculate the media and we have to give those cells time to grow and then do their metabolic pathway. When we take it out of the incubator, the first thing we're going to add is those 2 reagents. Now, I'm not asking you to learn the chemical names of these reagents because nobody learns this. Everybody refers to these as A and B or 1 and 2. Everybody in microbiology labs just calls these A and B or 1 and 2. So I'm going to add those 2 reagents into the tube. Remember, we put nitrate into the medium. And if the microbe that's growing in that tube reduces that nitrate, remember, reduction means a gain of electrons. If it reduces the nitrate to nitrite, I'm going to get a red color because those 2 reagents will tell me that there's nitrite in the tube. Those 2 reagents react with nitrite and produce a red color. That's a positive result. That tells me that that bacterium reduced the nitrate to nitrite. Now, if there's no red color after I add A and B, there's still 2 possible explanations. The first explanation is that the original nitrate is still in the tube. In other words, the bacterium is not able to reduce nitrate. That's a possible explanation. But the second explanation is that the bacterium can reduce nitrate. It's just that it further reduces it. It takes the nitrate to nitrite, and then it takes the nitrite to nitrous oxide, or maybe all the way to nitrogen gas. I don't know, but those are explanations. Either the organism can't reduce nitrate, or it can reduce it, it just reduces it further than nitrite. And how do we know which explanation is correct? Well, we keep going. We don't stop at that point. After we add the first 2 reagents, if the tube does not turn red, we keep going. The first thing we do is add the zinc. If nitrate is still in the tube, and that would be explanation 1, that the organism was unable to reduce nitrate. If that nitrate is still there, it will now become nitrite because zinc can reduce nitrate to nitrite. If nitrate is still in that tube, because the bacterium was unable to reduce it, the zinc will reduce it. And you're going to get a red color change because the zinc will take the nitrate and make nitrite. That is a negative test result because the microbe didn't reduce the nitrate, the zinc did. The zinc did it. Now, what if there's no red color after the zinc has been added? That means the second explanation is correct. The second explanation is that the microbe took the nitrate, reduced it to nitrite, and then further reduced it, either to nitrous oxide or to nitrogen gas. Either way, we've got a positive test, right? Either way, we've got a positive test. You have reduction going on. Nitrate was reduced. So you do have a positive test. But we want to know which thing happened. Is there nitrous oxide dissolved in that tube? Or was nitrogen gas liberated? How do we know? Well, we look at the Durham tube. There's going to be an upside down Durham tube inside our test tube. And we're going to look for a gas bubble. If there's a gas bubble in the Durham tube, then we know that the microbe reduced the nitrate all the way to nitrogen gas. If there's no gas bubble in the Durham tube, we know that the microbe reduced nitrate to nitrous oxide. Questions? Does this make sense to you? I know all the words sound the same. Nitrate, nitrite, I know. But does the concept make sense to you? The strangest thing about the nitrate reduction test is that the same color or absence of color can mean different things. Red can mean positive, red can mean negative, and colorless or no change can mean positive. That's the strangest thing of all of it. We have to add reagents in steps. We add A and B first, and then depending on what we get, we add zinc. And we got to look in that Durham tube and see if there's any gas in there. All right, let's do an experiment. Let's look at some bugs. We're going to use 4 different microbes today. We're going to use our old friend Staphylococcus epidermidis. Remember, this is a gram-positive coccus that lives on our skin, and some strains are pathogenic. Our old friend E. coli, gram-negative rod. E. coli lives in the intestine, which is why it's sometimes called a fecal or an enteric organism. E. coli is also a coliform because it can ferment lactose. We also have Pseudomonas aeruginosa. Remember, this is that organism we said was an opportunistic pathogen. This one is also a gram-negative rod, like E. coli. This one also has pathogenic strains. And this one can be found not only in and on our body, but out in the world. This one can be found out in wet environments and soil. It likes to live outside as well as on humans and other animals. We have a new organism today, though, and this one is Streptococcus pneumoniae. We talked a little bit about Streptococcus early on in the course, but we haven't seen a lot of strep in our experiments. Streptococcus is a gram-positive coccus, just like Staphylococcus is. Streptococcus pneumoniae lives in the respiratory tract in human beings, and there are some strains that are pathogenic. In fact, there are literally hundreds of strains of this organism. Imagine that. And most of them are pathogenic. Streptococcus pneumoniae causes what we refer to in medicine as pneumococcal disease. You may have heard this term, pneumococcal disease. Now, the pneumo here refers to pneumonia and the respiratory tract, and the coccal refers to the fact that this is a streptococcus. But despite the name, Streptococcus pneumoniae doesn't only cause pneumonia. It can also cause meningitis and septicemia. Technically, any bacterium can get into the bloodstream and cause septicemia, but pneumococcus or Streptococcus pneumoniae is notorious for doing this. This is a very dangerous pathogen. Look at those diseases. That's a group of diseases that nobody wants to have, right? We do have vaccine. We have vaccine against pneumococcal organisms like strep pneumoniae. We don't give them to everybody. Not everybody takes the pneumococcal pneumonia vaccine, at least not every adult does. We only give them to people who are at high risk of pneumococcal diseases. So yes, the older you get, you're more susceptible to pneumonia and specifically pneumococcal pneumonia. But there are other stages of life and other situations that people find themselves in where they may receive the pneumococcal vaccine. All right, so we've got this pneumococcal organism called Streptococcus pneumoniae. We've got Staph epidermidis, we've got E. coli, and we've got Pseudomonas. So we've got 4 very different sounding organisms. I just want to show you a couple of images this morning because pneumococcal disease really does have a very prominent place in human medicine. What you're looking at on this slide is a light microscope image taken from a patient who developed septicemia with Streptococcus pneumoniae. So what they did was they gathered a blood sample and they cultured the blood. And I want you to take a look. So we are under a 40x lens here on a light microscope. Check right here. See it? See that organism? It's stained dark, it stains purple with this particular stain that's being used. Here's a group of 2. Here's a group of 2. Here's this one that I pointed out earlier is 1, 2, 3, 4, 5, 6, maybe 7 cells. That is not something you want to see on a blood culture. You don't want to see any bacteria on a blood culture, but you especially don't want to see a pneumococcal organism on a blood culture. Interestingly, if you remember back to our discussion about gram staining, Streptococcus is a gram-positive coccus. It also has an arrangement, doesn't it? Streptococcus organisms like to form chains, and you can see that in this blood culture. It likes to form these little chains, and that is a characteristic of this organism, this genus. These bacteria like to chain up. Here's a nice long chain right there. So a bad pathogen, if you are unlucky enough to get it, which is why we developed a vaccine for it. Now, the next slide I just want to show you, I'm going to give you a warning because I don't usually make warnings, but the next slide is particularly gross. And if you'd rather not look at something particularly gross at 10 o'clock in the morning, you don't have to, you can turn away, okay? But I want to show you a picture of a brain that was taken from a patient who had pneumococcal meningitis. I want to show you what this organism can do to a human brain, okay? Look away if you need to, no shame. This brain was taken out of a meningitis patient. We are looking at the ventral surface of the brain. It's got this sort of brownish copper-colored tinge because there's still some meninges over the surface. And of course, there's blood circulation in meningeal tissue. This is what I want you to look at right here. You see that white area? That, my loves, is bacteria. That is pus. That is all pus on the bottom of that brain. This is what we call a suppurative meningitis because it produces all of that pussy, nasty material up around the brain. This person did not survive, of course. This is a very dangerous disease, pneumococcal meningitis. Over here, this is a sample of cerebrospinal fluid. It's very common when we suspect a meningitis to take a sample of cerebrospinal fluid. Now, what you're looking at here looks a little odd because it has a dark background. This is a very particular type of image. This was taken with a fluorescent microscope, but you'll notice that I don't see any bright colors here that I normally associate with a fluorescent scope. These particular cells, these are streptococcal cells, Streptococcus pneumoniae. They've been labeled with an antibody. What they did was they added antibodies into the CSF sample, and the antibodies had a fluorescent tag attached to them. So the white that you're seeing is actually light coming off of those cells under the fluorescent microscope. That's not nearly as important as noticing that these are bacterial cells. These are circular or spherical cocci, and this happens to be a Streptococcus pneumoniae. Now you'll notice that they're not really chaining in here. Remember, arrangements are good to see under the microscope, and they're good to notice, but you can't count on them. CSF, they don't tend to do it in CSF. They don't tend to form their nice chains in cerebrospinal fluid. But it's never normal to see bacteria in the CSF. So this patient was obviously very, very ill with meningitis. Okay, just a little, little medicine this morning. Little pus, in the morning. All right. Just as a reminder, again, I want to give you lots of tools in your notes for studying purposes. Here's a nice flow chart to help us read nitrate reduction results. First of all, over here on the right, this is what nitrate reduction broth looks like when it starts. So it's a very boring sort of beige color, typical nutrient broth. Remember, we supplement this broth with nitrate. This is what that red color looks like. So it's a very obvious color change to a bright cherry red. Remember, I'm going to inoculate this tube. I'm going to incubate for a full 48 hours. When I take the tube out, I'm going to add reagents A and B. And I'm looking for this. I'm looking for the red color change. This is a flow diagram to show us how we conduct the test. Now, this particular flow diagram names reagent A and B. Don't worry about these names. You don't need to know these for me. Just know that the first thing you're going to do is add reagents A and B. Now, if you see a red color, you've got a positive result. You can stop. You can stop the test because you know that there is nitrate reduced to nitrite in that tube. It's the presence of the nitrite that has turned the tube red with these reagents. Now, if I add the reagents and I don't see a color change, one of two things is happening. Either my organism is unable to reduce nitrate, or my organism is able to reduce it further than nitrite. So I'm going to add zinc. If the tube turns red, I've got a negative result. My microbe did not reduce the nitrate, but the zinc did. There must have been nitrate in that tube because zinc can reduce nitrate to nitrite. If I add zinc dust and it's still colorless, well, now I need to look into the Durham tube. I know I've got a positive test for nitrate reduction, but I don't know which end product was produced. So I'm going to look into the Durham tube. If there's a gas bubble in there, I know that my microbe took the nitrate and reduced it to nitrogen gas. If there's no gas bubble in there, my organism took the nitrate and reduced it to nitrous oxide. All right, hold on just one sec. I gotta back up. Oh no, I don't have what I need. Shoot, hold on. Hold on, everyone. Be patient with me for a sec. Be patient with me. I'm looking up something because I'm missing a couple of images I thought I had. Darn it. All right. All right, for some reason, I've lost a couple of my slides. Remember, we were also talking about the oxidase test. I wanted to show you a picture of an oxidase reaction. And for some reason, I've lost it out of my slide set. So I'm going to post it for us. I'm going to post it up on the discussion board, the picture. But remember, when we do the oxidase test, what we're going to do is take a cotton swab and we're going to pick up some colonies off a plate. You're then going to take a tube of what's called oxidase reagent, and you're going to drip that on top. You're looking for a color change. You're looking for purple here. If the swab turns purple, you have a positive test. And what that tells you is that your microbe contains an enzyme called cytochrome C oxidase. And that means something to us. It means that your microbe uses the C version of the cytochromes in its electron transport chain. That also tells us that the microbe also uses oxygen as its final electron acceptor. So that's a good piece of information. Now, there's a couple things about the oxidase test I want you to have in your notes, and this is much more practical than anything. The oxidase reagent, like all reagents, you buy from lab supply companies. It comes in a couple of different forms, strangely enough. You can buy little squeeze bottles of it. You can buy little single-use containers called ampules. And you can also buy strips of paper that have the reagent in them, like they've been impregnated with the reagent. Now, let me tell you a secret about the oxidase reagent. Once you open the bottle, if you buy that squeeze bottle, once you open the bottle and start using it, it expires really quickly. It expires within a couple weeks. So if you're a laboratory that does a lot of oxidase testing, that's not a problem. You're going to go through that little bottle quickly. The bottle typically contains like 15 mLs, maybe 20 mLs. It's a little squeeze bottle, like an eye drop bottle. So it's not a big deal that it's only going to last a couple weeks because you're going to use it. But if you're a laboratory that doesn't do a lot of oxidase testing, you don't want to buy that. Because as soon as you use it, as soon as you open it, it's expiring soon. It's going to expire within a couple weeks. It has to do with exposure to the air. It's only going to be good for a couple weeks once you open it. And if you're not going to use that whole bottle, you're going to have to throw it away, and that's a waste. So you might want to buy the test strips instead, because the test strips are individually packaged. So anytime you need to do an oxidase test, you just open up the test strips. And they're really easy to use. You take your cotton swab, you pick up a couple colonies of your organism off a plate, and then you just rub them onto the test strip. And what that's going to do, if it's positive, is turn purple. The test strip turns purple. It's got the oxidase reagent in it. the paper has been soaked in oxidase reagent. So you just rub the colony material onto the test strip and you look for a purple color change. That's a positive test result. And again, I will put a picture of this on our discussion board for us. If you take that colony material and you rub it onto the test strip and you get no color change, that's an oxidase negative result. That organism doesn't have the enzyme called cytochrome C oxidase. And that means it doesn't have cytochrome C in its electron transport chain. Now, here's a question. Here's a question. Does that mean it doesn't use cellular respiration? If it doesn't have that cytochrome C in its electron transport chain, does that mean it doesn't do cellular respiration? It doesn't have that little messenger to pass the electrons along. So maybe it doesn't even do. Is that how I should read that? If it's negative? If it comes up negative on oxidase, it doesn't have cytochrome C. Does that mean that it's not doing the electron transport chain? Does that mean it's not doing cellular respiration? What do you think? Carrie's got it. Connie's got it. No, remember, there are 4 kinds of cytochromes in bacteria. It just means it doesn't have the C version. It could have A or B or D. Don't forget that. Don't forget that. There are multiple cytochrome types in bacteria. They're very diverse. They're very diverse. All right, so I lost my pictures of the oxidase test, but I do have the results here. The Streptococcus pneumoniae organism will be negative on oxidase. The Staph epidermidis is negative. The E. coli is negative. But Pseudomonas will come up positive on this test. Pseudomonas, therefore, has the cytochrome C oxidase enzyme, which means it has the cytochrome C version in its electron transport chain, which also means it uses oxygen as its final electron acceptor. These guys, something else is happening. That's all we can say. That's all we can say. They may be using a different version of cytochrome. They may be doing a different pathway. We can't make any other conclusions about them. Now let's look at our nitrate reduction test. All right, take a look at this picture. You can see test tubes with our organisms in them. Let's start over here on the left. Here's Streptococcus pneumoniae. After 48 hours, we took it out of the incubator. We added reagents A and B, or what on this slide they're calling reagents 1 and 2. Here's the color. What do you think? Oh, sorry, I forgot to mention the most important part. When we took this tube out of the incubator, we added reagents A and B, or 1 and 2, and we got no color change. This color only happened after zinc was added. So I want you to think, is this a positive test or a negative test? And what do we know? How would we interpret that? Strep pneumoniae stayed colorless after we added reagents 1 and 2, or A and B. So we added some zinc dust in there, and we got that cherry red color. Is that a positive or a negative test? And how would you interpret it? What does it mean? Connie says nitrate reduction negative. Nitrate reduction negative. But it turned red, Connie. I don't understand. I thought red meant nitrite, and I thought, I don't understand. Isn't red a positive test? Good job, Elaine. Negative, because the microbe didn't reduce the nitrate, the zinc dust did. The zinc confirmed for us that the microbe was unable to do anything with that nitrate. It did not reduce that nitrate. The nitrate, in other words, was still in the tube 48 hours later. How do I know? Because zinc will reduce nitrate to nitrite. So if I get a red color after the zinc has been added, I've got a negative test result. The microbe is unable to reduce nitrate to nitrite. The reason it turned red is because zinc can do that. So the zinc did the reduction there, not the microbe. Good job. All right. Now look at the next two tubes. Staphylococcus epidermidis is in this tube. E. coli is in this tube. These two tubes came out of the incubator after 48 hours. Reagents A and B were put in, and they turned red. Remember, they're calling them N1 and N2 here. Don't worry about that. It's still A and B to us, or 1 and 2 to us. I got a red color after the first two reagents were added. How do I interpret that? What does that test tell me? Is it positive or negative? And what does it mean? What does it mean for Strep pneumoniae and E. coli that the tube turned red after I added A and B to it? Go ahead and type it right into chat. How would you interpret that? The tube turned red after I added that A and that B reagent to it. Good, good, good. Type it right into chat. Positive test. Yeah, that's nitrate reduction positive. The microbes in those tubes, the Strep pneumoniae and the E. coli, took the nitrate in the medium and reduced it to nitrite. I know because when I add reagents A and B, if there's any nitrite in that tube, the media is going to turn red. If there's any nitrite in the tube at that moment, the media is going to turn red. And it did. So the microbe reduced the nitrate to nitrite. Now, here's a sort of a technical question. How come there's two different colors? How come this tube, which I had to add A and B and then zinc, this tube is this color, and these two tubes are a different color? Why does that happen? I mean, they're all red, but why are there different colors of red, do you think? Any ideas? There's obviously indicator dye in here, right? What does indicator dye, what makes an indicator dye change color? What characteristic of the media makes an indicator dye change its color? What chemical feature? What characteristic? What is it about the media that will cause an indicator dye to shift color? Yeah, Carrie's got it. pH. pH. Anytime you put something into a tube, you're going to change its pH at least a little. When you put A and B in, you got that cherry color. Or I should say, it's not cherry, it's an orangey red, we'll call it. Then we added zinc dust to one of those tubes, and we got an even deeper red. Every time you add a different reagent in, you're going to change the pH a little bit, and that's going to shift the red a little bit. Just so you know, that's why sometimes at different steps in a complex test like this, at different points, that indicator dye is going to shift colors because it's based on pH. And anytime I add a new reagent into a tube, I'm shifting the pH a little. Yeah. So it's still, you still read them as red. You still read that as, whatever it means. But that's why you get that little bit of difference in the red color. All right, here's our last tube. Pseudomonas. I pulled the tube out. I put A and B in. Nothing happened. So I added some zinc, and it stayed colorless. It stayed the same color. What does this mean? I added A and B. Nothing happened. I got no shift in color. Then I added zinc dust. I got no shift in color. What does it mean? How should I interpret that test? Is that a positive test or a negative test? And what does it mean? How would I interpret that test? Is it positive or negative? That's the first question. Colorless, even after zinc was added. Positive or negative for the reduction of nitrate? Type it right into chat for me. I added A and B, no red. So I added zinc, still no red. Positive or negative? Carrie's got it. Connie's got it. Elaine's got it. It's a positive test. The nitrate was reduced to nitrite, yes. The reason it didn't turn red is because the organism then reduced to the nitrite. It kept going. And the only way I'm going to know how far it went is to look in the Durham tube. I need to look into the Durham tube. If there's a gas bubble in there, I know that the organism reduced the nitrate all the way to nitrogen gas. That's as far as you can reduce it. If there's no gas bubble, I know that the organism took the nitrate and reduced it to nitrous oxide. The reason there's no red color in this positive result is because there's no nitrite in the tube anymore. There was nitrite momentarily, but that nitrite was then further reduced. So it's gone now. That's why you didn't get the red color change, but it's still a positive result. And I have to look into my little Durham tube to know. I recognize it's very hard to see on this particular image, but if you look over here, this little thing right here is the Durham tube. And I think you can just see a liquid interface right there. There's a gas bubble in here. There's a gas bubble in there. So, one more thing to put in your notes here. Just a little pro tip on the nitrate reduction test. If you put your microbe into a nitrate reduction test tube and you put it into the incubator for 48 hours and you take it out of the incubator, little pro tip, if you already see that there's a gas bubble in the Durham tube, even before you add those first two reagents, you know you've got a positive result. You see that? If I pull my tube out, before I do anything, I see a gas bubble in the Durham tube. I already know my result. I've got a positive test for nitrate reduction, and my organism reduced it all the way to nitrogen gas. That's the only way I'd get a gas bubble in that tube. Now, as a good microbiology technician, I would still conduct the test. Of course I would, because I want to confirm it. So I would still do it. I would still add A and B. I would expect to see no color change. Then I would add my zinc powder and I would expect to see no color change. And now I have confirmed that my microbe took that nitrate molecule, reduced it to nitrite, but then further reduced it to nitrous oxide and then nitrogen gas. And I caught a little bit of the nitrogen gas in the Durham tube. So yeah, just a little pro tip there. You can look in your Durham tube right away and sometimes know what you've got before you start adding your reagents. All right. So the nitrate reduction test is perhaps the most complex test we do, at least in this course. And what makes it complex is that, first of all, it's complex to do. You have to add reagents in a particular order, in a particular sequence. But it's also very complex because you have to understand what's happening in the tube. And that's how I want you to study it. That's how I want you to outline it in your notes. I want you to be able to describe what's happening, why I add A and B. What are A and B going to show me? It's going to show me if there's any nitrite in the test tube. And what does that mean if there is nitrite? Well, it means that the organism took the nitrate and reduced it. The organism has the ability to do that. It has the enzymes necessary to reduce nitrate to nitrite. What about if I put A and B in and it stays colorless? What do I do next? What does that mean? What if it's still colorless? What does that mean? What do I do? How do I interpret it? So that's what you're going to want to do. You're going to want to use whatever slides you found helpful to make sure that you can walk and talk your way all the way through that test. Okay? Remember, anaerobic organisms that do anaerobic cellular respiration and oxidative phosphorylation, they don't have to use nitrate as their final electron acceptor. There are other molecules that will serve just as well. Sulfate, carbonate, those are common. And there are even others. It's just that nitrate is one of the top three. Nitrate's a very common one. So that's why it makes good sense to test for it in the lab. Because if I have an organism that's doing anaerobic cellular respiration and it's using nitrate as its final electron acceptor, the way it uses the nitrate might help me identify that organism. If I took all the different bacteria that were anaerobic and used nitrate as their final electron acceptor, some of them are going to reduce it to nitrite. Some of them are going to reduce it to nitrous oxide, and some of them are going to reduce it to nitrogen gas. So it helps me as I try to identify this bacterium. It helps me to know which one it's doing, because I can look that up. I can look up in a textbook. I can see the list. Which organisms take it all the way to nitrous oxide? Which ones take it all the way to nitrogen gas? And it will help me. It will help me in my quest to identify that organism. All right, guys, good job. So remember, you have actually 2 separate lab homework questions. They're not long. You have one for protein catabolism, and you have one for this material, this respiration material. Those are due Sunday by midnight, as always. Don't forget, you have that lecture quiz due Friday at midnight for immunology. Difficult lecture. Give it the time that it needs. Take as much time as you need to make sure that you get a nice, complete set of notes. It's a long lecture, which is why it's the only one we're doing this week. So get that quiz in by Friday at midnight. Get your lab homework in. When I see you on Monday, we're going to talk about your last assignment for lab. The unknown microbe. Identifying the unknown microbe. And of course, next Wednesday, a week from today, is your final lab practical. And it will cover everything we've done in lab since your last lab practical. So you have a week now to make sure your lab notes are all in order for that. Okay? All right. I'm going to post those oxidase pictures for you just so you can see them. I'll send out an announcement when that's done. Enjoy the rest of the day. Stay dry. I know a lot of us are dealing with a lot of rain right now, so stay in touch if you need anything, and I'll see you on Monday.